Right now, question is, what is the use of these stem cells? So we have embryonic stem cells, we have adult stem cells, and what is the use of these stem cells? Well, answer is like this. Still, we haven't understand this differentiation process. If we have a cell and we can differentiate into any kind of a cells, and we don't know exact process, it's very hard to use for full uh, in a wide range. Then we can use stem cell for research to understand the process. Second thing is, if we know a detailed mechanism of certain stem cells, how it work, we can use for clinical application. Example, we take a very simple example. If there is a damage to the body, and this damage is the tissues which is regenerate, tissues which has ability to regenerate, then the new cells are growing, then the damage is replaced. But certain tissues, it is not possible. We know largely nerve tissue cannot be regenerated, and also muscle tissues like a cardiomyocyte, heart muscle cells, it's very difficult to regenerate. So we can now find a method how we can still differentiate stem cell into nerve cells, the stem cells into muscle cells. So if we can achieve this differentiation, then we can use to treat diseases, or in a broad way we can grow organs. So if we have a stem cells, we use these stem cells to make organs, then we can use for organ transplantation. Right, before we understand a little bit more about the use of stem cells in the clinics, we're trying to see this video. Okay, do you see this video?
Yes, sir.
Okay. Okay, if there is a damage internally inside the body, and this tissue is has the capability to regenerate, then new cells come into this place and start to make new tissues. So generally, just to understand this, so the cells, many cells transport in the blood. So these cells transport everywhere in the body. When there's a damage, the chemicals are released. You can see the chemicals are released to the damaged area. And then these chemicals make new cells. Okay, here. Yes, this one. Now this one, if you look, what's going to happen, blood sends some chemical to the tissues, and this chemical stimulate these cells to divide, and they divide again, enter into the blood. So how this happen? The bone marrow has stem cells. Blood is produced, blood cells are produced in the bone marrow, and red blood cell and white blood cells continuously produce and release into the blood. But in certain situations, these cell numbers are increasing. For example, if there is an infection, white blood cell number increase. So when there is an injury, damage to the body, immune response is increased white blood cells. So how this happens? The chemical released from the blood to the bone marrow, and bone marrow, these chemicals, cells of the bone marrow take these chemicals and then start to increase their number, like this. Right now, if there is an injured tissue, they also secrete chemicals, and this chemical attract the cells from the blood. Here is the small chemicals moving to the blood, and they attract the stem cells into the tissues. And then stem cell can divide, and also they can differentiate, and they can replace the damaged tissue. Yes, Lucia. Okay. Now you understand the natural process is inside there are stem cells, and chemical released by the damaged tissue attract the stem cells, and stem cell can migrate to this place, and they can divide, differentiate, and they can heal the area. So there is a limitation. It cannot happen at always, and it's not happening at all the tissues. But if we can understand what are these chemicals and how this differentiation take place, scientists can still use stem cells for diseases like myocardial infarction, Alzheimer's, and some other diseases, brain diseases like degenerating disease. That means neurons number decreasing because they are dying. So if we can replace these neurons with the stem cells, then we can find the treatment. If the heart muscles died because of ischemia, myocardial infarction, then we can inject these stem cells, and stem cell go into that place and replace the damaged area. But this is not a very easy process, but we are one step forward with some of the treatment. So what is the well-known treatment? If we take a bone marrow, the bone marrow has a stem cell. Right, now this bone marrow can make white blood cell and red blood cell. On the other hand, there is a negative side of this. If this stem cell mutated, they make the cells, but these cells are not the correct cells. And we have learned cell cycle. The cancer situation is it lost the regulation. Even stem cells are under the regulation. They are dividing, but cell cycle is controlled by these genes like proto-oncogenes and tumor suppressor genes. So their cell cycle is under control. If there is a mutation of these P53 proto-oncogenes and tumor suppressor genes, then they lost the control. Then stem cells try to divide and produce more and more cells. So when they speed up the process, they cannot produce a correct cells because it's an uncontrolled process, and they are producing cells which are more and which is not in the correct, they are not in correct format. Their function is altered and the cells are not right, number is high. If this happens, there is a common situation. It's called leukemia. It's a blood cancer. So leukemia is a blood cancer. And the person develops a large amount of white blood cells, and the white blood cells are not properly functioning. As a result, their immune system is weaker. These cells cannot protect the person. These cells cannot function properly. And the problem is stem cells in bone marrow has a mutation. Right now, how we can use stem cell therapy to treat a leukemia is this. Okay, so we can find solution. If you really want to find a good solution, we should completely remove these cells. We should remove these stem cells which has the mutation. Then we should replace a correct cells which do not have a mutation. That is our way of thinking. If we have to find a solution, we need to remove these cells because they are the defective cells. If we remove these cells, we want to bring new stem cells. They are not defective. Right, we'll have this the stem cell therapy video, bone marrow transplantation, and we'll see it now. This is the bone marrow transplantation center, and usually the process starts with the patients, doctors, healthcare workers, the meeting, and then explaining them how it works and signing the documents and understanding the protocols. So it's a inpatient education program. So doctor is explaining what is this bone marrow transplantation, and the patient is get this information. Then the patient can agree to do the process, because in medicine, there is a strong concept called patient autonomy. That means no one can decide whether the treatment should be done or not. Only the patient can decide. So doctors can't force patient to do the treatment. Doctor's duty is, look, there is a treatment like this. If you would like, you can use it. It's effective, it works like this way, and you can use this treatment. The patient can reject, patient can accept. That is a patient autonomy. Even the patient's relatives, they can't force. It's only the patient decision. After that, patient signs the document and they can start into the process. Right now, there are basic tests they have to do. If the healthy cells are taken from another person, they want to little bit check that person's the cells are healthy and they are free of diseases, and also at least matching with the other person. So that's called tissue matching. So it's broadly the genes and the antigens like things, how far they are close to each other. These they are doing these tests. After that, they have to take, I think if you look, probably she's a mother or relatives of this patient, and you can see they are taking blood from the donor. So this person willing to provide the blood to the patient, and they're taking the blood from the donor. Say also they collect the stem cells from the bones, and then when they collect the stem cells, they can store stem cells in frozen conditions. The frozen condition is minus 196 around, that is liquid nitrogen. So minus 192, that is liquid nitrogen. We can store them on the liquid nitrogen and cells are viable. And after that, the patient has to undergo a very special treatment called radiotherapy. So radiotherapy is the treatment. They're going to target the bone marrow stem cell of this patient. So this radiotherapy can destroy the defected bone marrow cells. So they know exactly what is the radiation they want to use and which area they have to target, and they more focus on the bone marrow, and then radiotherapy treatment destroy the defective cells. So they can take this frozen stem cell now from the liquid nitrogen, and they can transplant these frozen stem cell after their thawing. They can again inject into the patient. So now the patient is getting donor bone marrow cells from from the donor, and they mix this bone marrow with the blood. So now the patient got this new bone marrow cells after the radiotherapy. So she has to stay inside the hospitals until the bone marrow cells completely move into the bone and also completely attached to the bone marrow, and they start to make new cells. During this period, the immune system is weaker, so they have to keep a little bit inside the hospital, a little bit careful. They have to take a rest during this period, and after that, once this stem cell completely going to the bone marrow, and they produce new cells. They can check by another test whether the patient is producing right cells and their number of white blood cells are correct and ready to dispatch from the hospitals. Then the immune system is again restored. So you can see they're checking the doctor final release from the hospital, and the white blood cells count is correct, and she's making right cells, and she has no defective cells now. Yes, this is one successful treatment doing at the moment. Okay. So that's it about the stem cell and use of stem cells. So if you go to the note section, you can see the exactly what is definition for this pluripotent, totipotent, multipotent stem cells, their location and their use. Right, the last lesson for these stem cells, we should also consider using bone marrow has ethical issues. Not the bone marrow, directly the stem cells. Because if we use stem cells from the embryonic stem cells, there is a big ethical issue. This embryo has life. Again, as you learn from the genetic test, the embryo has a life and embryo has a right to live. So then largely use of embryonic stem cell is unethical or even illegal. But some countries, they now are using adult stem cells, and also some of the countries, what they do, when the baby is born, the small piece of the connection between mother and baby is called umbilical cord. They actually froze this umbilical cord. So they freeze this umbilical cord, and the frozen umbilical cord is helpful to get some stem cells. When this person encountered leukemia-like condition, then this frozen umbilical cord can be used to isolate stem cells. Then they can use this stem cell to inject that person. It is much better than giving a stem cell from another person, because when we take stem cell from another person, they have some antigens which is different. So then there is a chance of rejecting this stem cell. They have to give immunosuppressive treatment to suppress the immune system. But some countries, they keep this frozen umbilical cord to isolate the stem cells so they can use it in the future. And bone marrow transplantation is a very successful treatment for leukemia, and many countries they are practicing this method. Okay, so we have done stem cell section. The last lesson is gene expression control and differential gene expression. Right, that section actually looking what is exact process. That means when we are trying to explain if these cells continuously divide and come into one stage, and after that differentiation start. So what is the mechanism of differentiation? Who is deciding which cell should go into this, which cell should go into the other side? So we're going to understand this process of differentiation, and after that we should also understand one cell like red blood cell differentiated, and one cell differentiated into nerve cells, and one cell differentiated into muscle cells. except for the red blood cells. They have a nucleus. Here they have a nucleus. This nucleus has complete number of chromosomes, and this original cell also has complete number of chromosomes. Then we have a question: if they have a complete number of chromosomes, if they have a same gene, this cell, and these cells also have the same number of chromosomes, same number of gene, and why they behave differently then? If their genetic information is exactly same, what decide these differences? Why a neuron is different shape and why it is has different function? Why muscle cells has different shape? Why muscle cells has different function? So we can understand maybe this information stored inside the DNA, and the DNA deciding what exactly the cell should do. Then if this cell has genes, some genes are active, some genes are not active when it comes to these cells. Otherwise, all the genes doing same way, then it is not the specialized cells. And all the genes are in a very constant stage, then just like a stem cell. So we can understand then some of the genes are very active and some of the genes are not active. We call this switch on and switch off. So this all the genes, some are going to shut down the process of activation, some going to activate. Then the activated genes can dominate in the neuron or muscle cells. Rest of the genes silence throughout the life. So now we come into understanding a little bit. There is a mechanism. This mechanism completely stops some of the genes, and there is a mechanism that completely activates the other genes. So when the genes can express, that means activate differently, then it can produce the effect. So that's we're going to learn under differential gene expression. Differential gene expression. I think next lesson we're going to learn this differential gene expression and molecular mechanism, and eukaryotic and prokaryotic gene expression a little bit, and also what are the factors which can affect gene expression, the environmental effect, and after that we can finish this section. So probably next week we can finish topic 3C, and we're going to start the next lesson. We can speed up the last part because it is plant. First section is about the plant. Second section is the environment and biodiversity. So there are a little bit of familiar topics, except certain things. We can quickly go through this section. Hopefully August we can finish, and then we can start doing papers after that. So I think you have to now start revising the lesson. So far we did. Once we start the lessons, papers, it helps to answer easily the questions if you remember the basic concept of things we learn. Yes, do you have any question? There's one, Sanjun, you have one question, right? Okay, there's a question. Can we create organs from stem cells? It is mostly under research now, because stem cell research is a promising area, but many scientists are struggling a lot with stem cell research because it's a very difficult area and so much of information we don't know. I mean, if you look at this picture, what make these changes? We know that it's changing to these cells. So what kind of a thing make the changes? So if you have a zygote and zygote develop into small embryo, and if it is so easy to give these factors, we can develop a complete baby inside an incubator. So what are these factors? Is these factors completely coming from these cells? Is these factors coming from the mother's side? Is these factors coming from the environment? Is these factors coming from other source? We don't know much about this information. So if the scientists want to develop an organ inside the lab like liver, starting point is, okay, we take this stem cell, because this embryonic stem cell make a liver. When they're developing into embryo, they make a liver. If it is the case, then we can also do in the lab. But what are the factors we should provide? What are the conditions we should provide? It's quite difficult to make all this information at the moment. Mostly under research, one by one, scientists trying to achieve the story. For example, they use stem cells to make muscle cells of the heart. So if they can successfully develop muscle cells, then muscle cells can add on to the heart when there is a myocardial infarction. So this area regenerate, and then the heart, the left ventricle is functioning again, and there's no heart failure. So if they can achieve at least this kind of developing right cells and treat these cells for brain diseases or heart diseases, I think the next stage, complicated structures like organs can be grown. Yes, around the world, a lot of scientists doing research with the stem cells to make new achievements, and as a result of that, one of the scientists make a solution that they take this kind of a cells and they try to reverse these cells to make stem cells. So it's called inducible pluripotent stem cell, IPS, like inducible pluripotent stem cells. So if we have a problem with the embryonic stem cells, they use existing cells to bring it to stem cells, and after that this stem cell can be used to make any cells. So you need to know basic knowledge of definition of these three stem cells, and what is the use of these stem cells, and what is at least one or two ethical issues of stem cells. Okay, I'm going to stop. Probably next time also, due to the situation, we're going to have an online class. So go through the notes, read it, increase your revision now because exam are coming up. Then we can smoothly start the papers. Alright, so I'm going to stop here and see you again next time.
Thank you, sir.
Okay, bye.
Thank you for
Bye.
that.
Thank you.